And genetic recombination, we talked about this yesterday. This was the production of offspring with combinations of traits that are different from either parent. This causes the offspring to have non-parental phenotypes. And we saw that yesterday at the end of the day when we had the vestigial wings but with the gray body. Normal body color but awkward wing shape. You can have non-parental phenotypes. You can have unique combinations. These are recombinants. So a recombinant chromosome would be one that is appearing different than either which parent. And why does this happen? Right. And when does that happen? In meiosis 1 specifically? Prophase 1. If you have genes on different chromosomes, totally different chromosomes, there is a 50% chance or frequency of recombination. And do you understand why it's 50% chance of recombination if they're on two separate chromosomes? When you're doing meiosis 1 and 2 and you're splitting up those homologous pairs and those sister chromatids, remember how we have said it's 50-50 chance if it goes to the left cell or the right cell? Right? When you do independent assortment and segregation, the chromosomes will move to opposite ends, right? And they'll do so without any problem. There won't be no problem with that. That 50% chance of recombination will occur because it's 50% chance they'll go to the left side, 50% chance they're going to go to the right side. Hopefully you see that, and if you don't, I strongly encourage you to review the concept of meiosis and how chromosomes are moving in meiosis. Now along the way, we continue the work of THM. Do you know who that is? Thomas Hunt Morgan. Very good. And what he discovered is he discovered that some genes are linked. But the linkage was incomplete. So sometimes there was a link and sometimes there wasn't. If they're linked, that means that when the crossing over happens, they get linked, they get pulled as a combination, right? I used a key example yesterday a lot. These are linked these sets of keys, they're pretty close together, and these are linked. It's kind of like I've got my school life and I've got my personal life. If you get a school key, you grab a school key, it's likely that you will grab some other school keys, right? You grab a personal key and it's likely you will grab other personal keys, right? The degree of linkage is going to be directly proportional to how close they are, right? They would be more likely to be linked up if they were close together. So what we developed is we were able to construct from this data, so linkage percentages, allowed us to construct a genetic map, which is basically an ordered list of the genes on a chromosome. And the theory is that the farther apart two genes are, the higher the probability that a crossover will occur between them. Therefore, they have a higher rate of recombination. Mm-hmm. See, if they're close together, they will be linked together, right? But when you make recombinants, you're making unique mixtures, right? Do you see how it's a little backwards? And this is one of the struggles that I can't really teach. You just have to see it. I can't implant the picture in your head. But you have to understand that if they're close together, then they would be inherited together. But if you want to make recombinant chromosomes, then you would be looking for things that are probably far apart. Further apart, higher degrees of recombination.
And different things, like in one person have certain things that are far apart, and another person has totally different things that are far apart?
Unlikely, no. The specific loci for specific genes are going to be very much close to in the same spot. So, like when we do crossing over and we make recombinant chromosomes, as we say, for everyone, we seemingly are making about the same recombinants. So, and if you're really thinking deep about this, that means to make recombinants basically means that, we have taught you that your chromosomes are distinctly your mom's chromosomes and your dad's chromosomes, right? And now I'm saying that when you go off to make your children, your chromosomes of your mom and dad will hybridize. Does that make sense? So that your children aren't having exclusively chromosomes that are of your mom or your dad, but they're kind of in some cases hybrids of your mom and dad. And now we're just saying that this idea of recombination is really what that crossing over does. It creates. When we say you make mom and dad mixtured chromosomes, okay, now we can just say you make recombinants, because that's a more proper term probably to use. We can use a linkage map to show recombination frequencies.